everyone. In this video we are going to discuss on mutations in the DNA. What are mutations? Mutations are sudden changes in the nucleotide sequences of DNA, to be simple, in the gene sequences. Mutations are the major causes of various genetic disorders following either autosomal dominant or recessive or X-linked dominant or recessive patterns of inheritance. It is the type of mutation and the severity of the damage occurring to the gene which determines the type of inheritance the genetic disorder should follow. There are 4 types of mutations which causes mutations in the DNA, which are silent, missense, nonsense, frameshift. The first 2 types of mutations are less severe, which are silent, missense, compared to the nonsense, frameshift. For point mutations, so the first 3 mutations indicated here or mentioned here, those are silent, missense, nonsense, are point type of mutations. They are due to the change in the single nucleotide point or position within the nucleotide sequence, which can be either transition, that is purine to purine change. Purine is changed to another purine, that is adenine to guanine or vice versa, or pyrimidine to pyrimidine, that is cytosine to thymidine or vice versa. Next is transversion. Transversion is purine to pyrimidine. The nucleotide purine is changed to the nucleotide pyrimidine, that is adenine to thymidine or pyrimidine to purine nucleotide change. So these are the 2 possible causes of point mutations causing silent, missense and nonsense mutations. Let's see what is silent mutation. Silent mutation is silent, that's why it is called silent mutation. Why it is silent? Because as we all know, the amino acids are coding by more than one genetic code. So when the nucleotide mutation or the nucleotide substitution taking place in the 3rd position of the codon still encodes the same amino acid, that does not change the amino acid composition of the protein. That does not change the amino acid composition of the protein. So that is why it is called silent mutation. Even though there is a change or mutation, that is still silent because it is not altering protein structure or function. It still remains the same. Next one is missense mutation. It is named missense because the protein formed due to the mutation has lost the sense or missing the sense from the normal protein structure and function. The protein formed from the gene which underwent missense mutation has missed the sense compared to the normal protein. That is nucleotide substitution resulting into the changed amino acid. Here the amino acid is changed to another amino acid, and amino acid chemical property of the amino acids also matters. If the amino acid is changed to amino acid which have different chemical structure and chemical nature, it will alter the structure of the protein as well as function of the protein. That is non-conservative substitution of amino acid. What is non-conservative substitution? Non-conservative substitution means if the acidic amino acid in the protein is replaced by either non-polar or basic amino acid, that is called non-conservative substitution, which brings the changes in the structure and function of the protein. The common example is sickle cell disease. In the beta chain of the hemoglobin, especially at the 6th position of the beta chain, we generally find glutamic acid. But due to the missense mutation, the amino acid glutamic acid is changed to the valine, which has different polarity. So that changes the structure and function of the hemoglobin and changes the structure of the RBC from biconcave to the sickle cell, and changes the function of the hemoglobin by decreasing the oxygen affinity to the hemoglobin. So missense mutation is altering the sense of the protein encoded by the gene underwent or undergone missense mutation. Example is sickle cell disease. The next type is nonsense. Here, due to the nucleotide position change or substitution, the genetic code is changed to stop codon. So the translation stops abruptly in between. So it results non-functional protein. So it is very severe because the lack of protein may disturb the cell function. Next type of mutation is frame shift. Here the frame of the genetic codons is changed. The frame of the genetic codons is changed due to the deletion of one of the nucleotide or incorporation of the nucleotide into the genetic codon or nucleotide sequences. It has changed the frame of the genetic codon. So due to the change in the frame of the genetic codons, now the protein may be shorter or longer, or because the amino acid composition is changed. Because of change in the frame, the genetic codons are changed now. So completely a different protein is made, maybe shorter or longer protein, which has function altered or disrupted function due to the frame shift mutation. The common diseases which we come across are Duchenne's muscular dystrophy due to the defect in dystrophin gene, which is X-linked recessive disorder, and Tay-Sachs disease, which is autosomal recessive disease. The next type of mutation is occurring at the splicing site between the exon and intron, that is splice site mutation. So due to the mutation at the splicing site, now the spliceosome is unable to recognize the splice site and it retains the intron with the exons. So that will change the protein formed with altered function that may lead to cancers, dementia, epilepsy, some types of beta thalassemia.